lectures as I planned. And then what I'll do is we leave today, my wife and I leave today for Pune and 30th, yeah, I think, I don't know, 30th, is that right? And so the next session that I'll do will be this one, which will be on the 31st. So you don't have any reading for this, okay? But I would really encourage you to go ahead and start reading in my book. A lot of material, a lot of detail, okay? It's never going to be a bestseller, and that's not actually why I wrote it, although I wouldn't mind. But when we get back on the 31st, so I lecture to you on giving you an introduction once again on these particular topics. And then the next session, okay, we will look at chapters 1 through 3, the second session after I get back from Pune. We'll go through chapters 1 through 3, then the next to the last session, chapters 4 through 6, and then we'll do 7 and 8 on the very last session, okay? Some of those I have some PowerPoints that I'll give you as an introductory or supplementary if if our discussion sort of lags. But some of them I don't have PowerPoints for at all. It's just us discussing. So plan as if that's what we're going to do. You read, have questions, write questions out on it. It's one of the best ways, you probably know this from your study habits already, but what I do when I read material, like when I was writing this book and I was doing about 200 papers, references a month, I would write myself either on the PDF, usually on the PDF, I would write questions to myself as I went through, questions I wanted to address when I was writing, but also questions you'll want to address and ask here and let us discuss, okay? So that's our schedule until the end. It's flying past, it's amazing. Already been in China for a month doing 4 of these, and now feels like I'm almost done with this, and I don't want to be yet. All right, so these are the 3 readings. Let's go ahead and see about questions, comments. I'll ask you a question to start it out. We'll start with Axel Meyer et al.'s paper. We'll move to Scott's and my paper, and then we'll talk a little bit about this, although this is something that we'll talk about a lot with my book material, and we talked about yesterday, but in a broader context, not just natural hybridization. Okay, so what did Axel et al., first of all, what organisms were they studying, and what were their conclusions from their studies? Whoops, that was wrong. Would be that one, that one. I know I'm giving it away, but what did they study, and why? Why were they interested in studying this group? What is significant? What is important about the group? What kind of organisms? Okay, cichlid fishes. I know, it's up there. Trying to get you going. Work you in slowly. I don't want to upset any of you. Okay, so cichlid fishes, where are they? Yeah, the Great Lakes. They're talking about Lake Victoria, but the East African Rift Lake system. Malawi, Victoria, etc. Right? So they're talking about Lake Victoria. So about how many species did they say were there, or divergent endemic forms? Yeah, in the hundreds. There are many more suggested. Lake Malawi now has, I don't know what the latest estimate is, 600 or something? Separate lineages, i.e. species. Okay? Some unnamed. The ones that are on my book cover, this new one, those are part of the Lake Malawi assemblage, one of them being a homoploid hybrid species of those fish. Okay, so Lake Victoria cichlid fishes. And from the title, but also from your reading, what were they testing? Okay, so you have 200 forms how could those, what are the alternative hypotheses about how those forms got to be in Lake Victoria? Let's talk about that first. What could have been the source and the origin of those? Okay, many different invasions. Simplistically, 200 lineages came in at once, but yeah, over the life of the lake, 200 lineages come in, or most of them come in, let's say. Where are they coming in from? Where are they originating from? Yeah, riverine habitats that surround and are all through this area, and actually throughout Africa, that region anyway of Africa, there are huge, many assemblages of riverine cichlids. We sort of think about them in the lakes, right? But the source of the lineages in the lakes are from the rivers. Okay? So, many invasions is one hypothesis. What's another hypothesis? Go ahead.
You can have one taxon breaking up into several of those one species.
Okay.
Or you can have polyphyletic if there's a hybridization, what we talked about in the
Okay.
thickness of the hybridization.
You're getting ahead of us. Stop. That's exactly right. So it could be polyphyletic in a lot of ways, right? In terms of those being there, they could come in already formed as individual species from the riverine habitat. You could have one form come in and speciate into, say, 5, and another form come in and speciate into 10. And so you could still have a polyphyletic origin for the entire species flock, right? Okay, what did Axel Meyer et al. Conclude? Yeah, monophyletic. All 200-ish arose in the lake itself from a single lineage. Okay? What data did they use to test for this? Mitochondrial DNA. What do we know about it? Okay, maternally and largely maternally inherited in animal systems. When I was in Kunming, China, teaching one of the 4 short courses that I did there were about 120 students-ish in the room. And I pointed out that it is not unlikely that out of our population, me and all the students there would not be unlikely to have someone who had at least paternal leakage, maybe, both paternal and maternal mitochondrial DNA there. So I just say that to remind you that, once again, nature is not simple. But it's largely in animals, most animals, not all animals, but most animals, it's mitochondrial DNA is maternally inherited. Okay? What else about mitochondrial DNA compared to nuclear DNA? Okay, it's not supposed to be under strong selection, which I disagree with vehemently since it's packed full of genes. But anyway, yes, that's another thing. It's has been considered in the past, especially a neutral molecule. Yeah. Higher mutation rate, particularly in things like the D loop, okay, origin of replication, okay, which is part of what they were looking at in some ways. Okay, what else about mitochondrial versus nuclear? By the way, it has a higher mutation rate than most of the nuclear genome, right? There are portions of the nuclear genome that are much higher mutation rate than the mitochondrial DNA. One that I worked on have worked on in both plants and animals, the ribosomal nuclear genes, not the genes themselves, but the intergenic spacers. So quote-unquote non-coding, but they're actually regulatory sequences. But also we have things that boost our immunity, right? A lot of alleles, so you have magnifying selection there to capture all those mutations. But largely, mitochondrial DNA accumulates mutations on average, more frequently, more rapidly than nuclear DNA. What else about mitochondrial DNA versus nuclear DNA? Something that's very important as well. Does it recombine? No. Now I give you the caveat. It largely doesn't recombine. Once again, if we had enough people in this room, we would say that it is within reasonable statistical probability that one of us would have mitochondrial DNA that did show some recombination events. Okay? Never a simple, completely black and white answer for any of this. Rules are there to be broken by nature, is what we basically say. However, largely it does not recombine. Okay. So, monophyletic origin for this entire cichlid assemblage. So what? That's interesting. But why is it? I will tell you that the conclusion of monophyletic origin here is wrong. It's wrong for every lake. Okay? And we'll talk about that. We'll bring up the point you made about hybridization again. It's wrong for every lake. However, I wish my name was on this paper because this paper overturned a number of paradigms regardless of the incorrectness of their monophyletic conclusion. But why would I say that? What's the big deal about 200 species arising in one lake? Okay,
There was no, first of all, there was no geographic isolation. They were all in the same lake.
Okay, so,
And also, lake was very young.
okay. Lake is very young, and actually, you know, they talk about how young it is, okay? It is much younger than Lake Malawi and Lake Victoria, etc. The last time they were dry, because these are shallowish lakes, the last time they were either completely dry or mostly dry was about 15,000 years ago. It's even younger than what they thought it was. The data that they were quoting has now been updated, and we now know that these lakes are actually much younger. So in 15,000 years, 200 species, 200 endemic forms arose. Regardless of how they arose, this tells us something about the pace of evolution, the potential pace of biodiversification. Okay? Doesn't always have to happen in 15,000 years. The point is it can. And your point now about they're in a single lake, and yet we see the origin of 200 species. Ish. There's more than that, apparently. And yet they're still, initially especially, within cruising distance of one another. They could actually mate with one another. And yet they sorted into obviously available niches. And obviously if there are really 200 plus different lineages, there are 200 different realized niches, most likely. So we're looking at massive environmental dependent selection and very strong episodic natural selection. Do you see why I'm saying that we actually was talking to Alasa last night and yesterday afternoon, and we were talking about the molecular clock and the hypotheses around that and all this, and we both agreed that you know, dating things on trees is very tricky to do. Both of us are worried about dates on trees, whether they happen to be ours or somebody else's. And so, but part of that real concern is that it's not metronomic. The accumulation of mutations and evolution does not appear to overall to go in those nice equivalent steps, but rather in bursts. Okay, and so dating, putting those metronomic mutation accumulation along a lineage, counting back and saying this is how many million years ago it was, is is problematic. Okay? All right. So, when Axel Meyer came to our university, this was published in 1990. We had him as a guest in 91. By the way, Axel and I are friends. I've spent a month in his lab. I'm not trying to trash him or anything like that. He and I have had this discussion before, and he understands and agrees with it, what I'm about to say. But when I asked him, okay, I didn't even think about it being hybrid populations starting this, okay? So, but when I asked him, are these intergressively hybridizing at all within Lake Victoria, he said no. No evidence of it. He was wrong. He was really wrong, okay? But why might he have concluded that using mitochondrial DNA? Because that's the information they had. Remember, this is 1990, right? And so it's not that they were stupid. It's not that they were lazy. It's not that they didn't want loci to analyze. We just didn't have the technology for it right then, right, that we do now. Okay? But why might mitochondrial DNA have been a bad marker for hybridization? Let's talk about that. Or chloroplast DNA if you're working on plants. Why might it be a bad marker, a bad tool to use alone? I'm not talking about using it in concert with nuclear, because that's fantastic, okay? That's a way that's very powerful. But alone, using cytoplasmic, either chloroplast DNA in plants or mitochondrial DNA in animals. Let's talk about that. Why would it not be useful necessarily to detect intergressive hybridization or homoploid hybrid speciation? Okay, so it's from one parent, but let's unpack this for people who might not have thought a lot about this, okay? And because I'll give you another example of how this misled us in another group, i.e. our species, in a moment. But so it's one parent, so you don't capture any information from dad, okay, from the male lineage. And it doesn't recombine. But still, what if we have multiple, which they did, they had multiple haplotypes. Why wouldn't that tell them that maybe there was intergressive hybridization there? Yeah, right? I mean, since it doesn't recombine, you're not going to see hets. You're not going to see recombinants, right, from divergent lineages together, like you can with nuclear genomic material. So when you get so let's say you go out into an area and you're working on plants like we do, and which we do, and we go out and we look at the chloroplast DNA variation because our species differ for cytoplasmic. They have different haplotypes for our different species, okay, fixed differences. And we go out into an area where we see a red-flowered and a lavender-flowered species, and they're together. Now, they're not together right in the same niche, but they're close enough where pollen can be transferred. And we find, let me back up. We don't know if they're red and blue flowered, but they have these plants that are scattered around throughout this habitat. They haven't flowered yet, which is quite common. We take leaves, we haplotype them, and we find out there's an admixture of chloroplast DNA types. Is it a hybrid zone? We don't know. It may just be an overlap zone between the two species, a Walland effect, if you will, where we have a mixture of those haplotypes growing in the same area, but they're not exchanging genes. Axel's et al.'s study immediately suffered from that limitation. They could not test for intergressive hybridization, really, and they could not test for any kind of a non-monophyletic origin due to genetic admixture. But the reason for them it wasn't an issue is because in 1990, when this was put out, quote unquote, especially evolutionary zoologists knew that intergressive hybridization was not important. It didn't happen in animals. quote unquote. And if it did, it was minor, and there just wasn't a big deal about having to worry about it. Okay? So they weren't looking for it. They weren't worrying that they were going to miss that by using the mitochondrial. They knew that the mitochondrial DNA didn't show recombinance. They weren't stupid, but they didn't think they had to use, even if they could have, they wouldn't have worried about trying to use nuclear markers. For that reason, anyway. Does that make sense? Okay, fast forward to today. It's now known that and I've mentioned this, every one of the species flocks in every lake was begun, apparently, from a hybrid swarm. Yes? Say, go back to...
The part about having separate haplotypes.
Yeah. They're separate haplotypes. Okay? Is that what you mean? Or how do we know that there are separate haplotypes? Or...
And how does that not answer the
Yeah, so basically what they had is when they went in there, they found these independent lineages with these, I don't know if this is going to answer your question, with these kinds of separate haplotypes from very different clades, but they all coalesced to a single monophyletic origin, which is exactly what you would expect mitochondrial DNA to do. Now, let me mention that there are ways of analyzing these data now, and even back then there were ways. But I would argue time. But they had very little sequence data, and it came from a non-recombining molecule. Okay. So, like I said, we fast forward to today, present time, Lake Victoria, Lake Malawi, etc., Lake Tanganyika, the cichlid fishes, the flocks, those highly diverse species flocks, adaptive radiations, each one of them, when you trace it back, is non-monophyletic. Okay? And what we mean by that, remember, is not that multiple, this is where it gets a little tricky. They're 15,000 years old in Lake Malawi. We have, I don't know, 400, let's say 450 species. That was one value I remember from a way back. But I think they can count more now, but that's not an issue. 450 in 15,000 years is incredible. So 450 species or 200-ish species. And what we mean by non-monophyletic is not that we had, once again, not that we had 200 dump in there. Okay, 200 separate lineages that were already formed. What we mean is that this starting material leading to, say, 450 species, that starting material came from hybridization between riverine forms. And then a hybrid population, what Ola Seehausen and, well, he's quoting the terminology from much earlier in plant literature, actually. A hybrid swarm, i.e., phenotypically and genotypically, genomically, incredibly variable. Okay, this hybrid swarm, that much more variation than either one of those species, is actually what's introduced into the lake. So it's demonstrably non-monophyletic. It's like an allopolyploid is a non-monophyletic event. An allopolyploid polyploid speciation event is a non-monophyletic formation. Comes from two. Okay, two lineages. So this is the starting material. Okay, for each one of these lakes. On top of that, in addition to that, you have more recent, ongoing intergressive hybridization among the various forms, where basically you have a syngamion again. Remember that circle of, in this case, it would be hundreds of individual lineages that are assortatively mating within their own lineage. You still have linkage disequilibrium built up, right? So you're keeping the color variation, the jaw formation, you know, the architecture, whatever other adaptations are there, they're keeping together, but they're also exchanging genes, some of which appear adaptive. Okay? So that's more recent. So formed from intergressive hybridization and a hybrid swarm, leading to 450 species with additional more recent. It can't be too much more recent because we're talking about such a compressed time. But anyway, now let me reiterate something. The fact that it's a hybrid swarm does not mean that the hybrid swarm condition caused the adaptive radiation. Knowing this is really, I would, suggestive information for why you would be able to explosively radiate. But knowing this is not sufficient for concluding that hybridization admixture drove the adaptive radiation. You have to know other things. Particularly, you have to know that in this, be able to infer in this, that you had alleles that were adaptive or that could drive biodiversification that sorted themselves out in these different lineages, okay, into different habitats or different sexual selection regimes or whatever. You need to demonstrate something like that to actually have a much stronger inference. But it is highly suggestive, and it's cool to me that each one of the lakes was started in this way. That comparative data gives us some sort of a hint that maybe that was a key step for this very compressed adaptive radiation in each one of these lakes. But there are many other I've mentioned this already in the class, many other experimental as well as looking at genomic information, looking at the fish in the lakes, how they behave, blah, 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 whatever it happens to be. Many other steps to say anything, actually, ignore admixture for a moment, to find out what was the key step or the key process for an adaptive radiation is incredibly difficult, okay, to get a really strong inference. Okay, and we'll talk about, you know, when we go into Scott's and my paper, really should just say Scott's paper, because he was really the generator of all that stuff. He was just kind to let me work a little bit with him. But anyway, Scott's work on the columbines, we'll talk about that. The difficulty we had in designing experiments and also other ways of analyzing to see what might have caused, what may have been the causal factor for that adaptive radiation. Okay, questions or comments about Axel Meyer's paper? Yes?
In order for there to be so many different species, there would have to be those many different niches also.
Have to be,
Those many different niches.
that's the argument. Yeah. So remember, though, that when we're talking about niche differentiation, she's the one you need to ask about this, because she's the ecologist, okay? And so if I say something really stupid, you need to seriously correct me. But niche differentiation for us, it doesn't have to be that the niches don't overlap. It's just that they cannot overlap completely. So in this lake, you have casts, if you will, of certain kind of feeding mechanisms. Those that eat other fish, but still look quite different from one another. those that actually pluck the eyes out of other fish. I mean, it's just, that's their jaw articulation gives them, and their teeth formation or their protrusions in their mouth give them that ability. Scraping the algae off of rocks, another feeding. So, but you have whole clades of those kinds of fish scattered around the lake, right? So they overlap, but yet not completely. And so niche differentiation, I mean, these theories about tropical versus subtropical versus temperate, how many niches are there? That's really what we're talking. How are they packed together? It's a bit circular, but what we say is, hey, there's 450 different kinds there, so there must be 450 different niches. I know, that's probably not, yeah. But we can actually, they actually go out and they say, well, what kind of niches would there, can we subdivide this?
Is there any evidence that in many of these lakes that these different forms are actually competing with each other, or what are happening to their population dynamics basically?
Yeah.
So it may not be that there are 450 different niches. It may be that there are much fewer niches, but ultimately they will sort out and some will sort of decline.
So that is an excellent point too, that maybe there aren't 450 niches, maybe they are competing with one another. The answer is yes. They have good evidence that certain lineages do. The interesting thing is, though, that the lineages that would be most similar, we would guess most similar in their niches, are displaced in different places in the lake. Okay? And so it appears that in that case, they may have sorted themselves out into different areas. I mean, we assume that lineages went extinct, right? Even in 15,000 years, obviously, that's quite a few generations. So we're assuming that 450 is not the actual number. And so I would assume that competition, direct competition, led to displacement and extinction as well. That part of it I'm not certain. But what we can say, and let me hasten to add, is that in these complexes, what is thought, remember I told you about the wolves that are black? The same kind of a process is thought to be largely responsible for the diversification, and that is sexual selection. Female choice is supposed to be, no, female choice is known to be a very, very strong component in determining assortative mating in these. And so it's thought, I don't know, really honestly, female choice is sexual selection, but I always also think about it as ecological niche, part of the niche, because males are a certain color and it draws them to, anyway. But for whatever reason, we may actually have a trade-off as well where they can actually compete somewhat, but females are still choosing differently, and so they can actually continue to reproduce even with that competition. So there may be a balance, is what people have also suggested. Yeah, but great point.
So I realized that the paper was published in 1990, but how do you make these sort of conclusions, which is 14 lineages when there were actually 200 known lineages from the lake?
Yeah.
This kind of
Go ahead.
short sampling would, I mean, nowadays you need very, very comprehensive sampling to make these kind of conclusions.
Yeah, so the question is, maybe the bigger question is, but still I'd want my name on it, because they were right in terms of explosive radiation in a very short period of time rather than coming in from having 200 dropped into the lake. How could they have made the conclusions or felt like their conclusions were strong inferences based on such a small percentage of what was there with very little sequence information? And I mean, the short answer is that's a huge amount of sequence information for 1990. I did this kind of sequencing for my PhD, and to get, it's unbelievably, it takes a long time. And so it's a lot of information, but also keep in mind that what they were assuming was that they were going to pick out Yeah, and they were just they were choosing samples to reflect the diversity of that assemblage, right? They were choosing fish from what we were already thought to be very divergent lineages. And so what they were thinking was that if they chose fish that were more closely related to one another but still divergent from other fish that were very divergent from that first set, and et cetera, that if it was polyphyletic, they were not going to ever coalesce into a single root. And so that was the other sort of litmus test, was they actually were wise in the well, apparently, because I've looked to see where these fish are on this super tree that they have for all these species, and they are very divergent. You know, they're in different clades. So their idea was you subsample well enough across those divergent clades, and they didn't have that phylogeny then, right? They just had a taxonomy that told them what to select, suggested. So anyway, that's why they did it. Yes, nowadays we understand the kind of homoplasy that can be there. You know, the kind of, well, the amount of incomplete lineage sorting, intergressive hybridization, parallelisms, convergences, whatever else that go into that homoplasy estimate, you know, the estimate of experimental error, basically, for phylogeny reconstruction. And so we understand the degree to which things can be off because of that, and that we need incredibly complete sampling to draw phylogenies, per se, because we already know we're in trouble because of all the extinct lineages we're not sampling. And so we even, you know, we could never know those really. So yeah, you're right. Very poor sampling, very little sequence for what we now know. So it's surprising that they actually got it to where they did get it. Yeah. Yeah.
So if the parental lineage was not a hybrid, would you still have expected this sort of variation? And if it did, would you still call it monophyletic if there was subsequent hybridization efforts?
Yeah, so the question is, if you guys didn't hear, so if there wasn't an admixture event at the base of these, would we expect in very short period, let's just say over a very short period of time, relative number of generations, very few, would we expect that kind of explosive radiation? And if the second question was, okay, so you start with no admixture, and you're having this diversification, and they're intergressively hybridizing, though, now as they diverge, is it still monophyletic? Answering the second question first, yes, because it started with a single lineage. Now, it's some of these formations, let's say you have 15 homoploid hybrid species in the lake out of 200. Those are not monophyletic. But in terms of the entire assemblage, yes, we would consider it monophyletic. Would we expect this? If you had have asked me in 1991 when he visited, I had no clue. I mean, people were talking about adaptive radiations and had been forever, and we were working on columbines with Scott Hodges and looking at those, and we never considered hybridization as a driver. Scott really doesn't think about it that way, I don't believe, still to today. I think actually it may actually be the case in columbines, highly likely. But be that as it may, that's his system and he's working on it. But I don't know. I don't know. I find it now, with what I know about population genomics, et cetera, I find it very difficult to understand how you can get that kind of diversification out of a bottleneck, which is what that would have been. Without hybridization, it's a bottleneck. And so you have a few fish. We're thinking it's not 100,000 fish dumping into the lake all at once. You have a few fish, relatively small N sub E, from a single lineage, giving rise to 450 different lineages. The idea of the level of polymorphism that would have had to have been there in those, for the various adaptive alleles or alleles causing adaptations that we see in those fish or the sexual selection would have had to have been enormous. The subsampling would have had to have been, well, I just don't think a single lineage would have that amount, but please understand that we're very early days for testing any of the adaptive radiations, whether in cichlids or anything else, to see whether or not it could have been generated by admixture. They could have been caused by admixture. So I hope every freaking one of them was caused by admixture. But that is a real bias on my part. Okay? That is my bias. You all have biases too, but that is my bias. I really hope we find out that, yeah, you absolutely have to have admixture. You'll never get an adaptive radiation, especially over a short period of time like this. But I don't know. We don't know that yet. I'm just hopeful. I'm the hopeful monster guy. Yeah.
Yeah, I was wondering, has there been any studies so far on any of these snakes with this particular system, which estimates average divergence among the species?
That is, I'm sorry, I didn't hear the last part. That is what?
The measure of genetic distance or divergence.
Oh, divergence among the genetic divergence. Oh yeah, they have, like for Lake Malawi, they have blasted. Let's see, where did they publish the paper? Ola Seehausen actually published a paper, maybe in Molecular Ecology, where they have a phylogeny for everybody in Lake Malawi, and they used, I've forgotten how many hundreds or millions of SNPs. And so in those data, if you want to send me an email, if you want that, I'll look it up. It's, I don't know if it was genotyping by sequencing or not. It was like in 2013 or 14. I'll have to look it up. I've slept since I wrote that down. I write it in my book so I don't have to remember this crap. No, I don't remember that. I don't remember exactly which paper it was, but they were very cautious because they said, we got a resolved phylogeny using this many SNPs, and we think this is a really good way to do this, but we need some more information to make, because they know it's a hybrid swarm that they started out with, so they had to get through all the incomplete lineage sorting and all the genetic exchange events, both in the hybrid swarm that started it as well as since then, to sort their phylogeny out. We'll talk about testing between incomplete lineage, I know I've said this before, incomplete lineage sorting or retention of ancestral polymorphisms and genetic exchange, trying to determine if we can see how to test those alternative hypotheses to see what's causing our weird data, to not agree with one another. Like between morphology and genomics or something, that we have trees that don't agree. But what I will say is one of the most difficult kinds of data come from things like this. Very short periods of time since divergence, and as well, intergressive hybridization potentially going on. And the reason, short time since divergence, you don't expect coalescence to have been complete. And we'll talk about what that means when probably not, I don't know, a couple of lectures down, I'll show you a PowerPoint on incomplete lineage sorting and genetic exchange. We'll talk about how we might be able to discern between those two. But anyway, this is horrible. I mean, this is absolutely the worst possible because they arose, 450 species arose in 15,000 years, which means that all that allelic material that started out in the ancestor from hybridization or not was polymorphic. It's sorting itself out on these, and it could be genetic exchange, or it could, it's bad. Yes? Why what? Why couldn't it have been incomplete lineage sorting, you mean? Or polymorphism arising? Well, it can be. But are you asking why it might not be this? That it could just be incomplete lineage? So are you asking if it has to be hybridization? Yes, yes. It is sorting itself out from the, you mean these? Is that what you're, I'm not sure how far back you're going when you're calling it parental, but it is in these. In this? Yeah, this is a hybrid swarm which is generated from these, right? So the standing genetic variation in this riverine form and the standing genetic variation in this riverine form combined here. It wouldn't be all of it because you would just have a few individuals hybridizing or one or two of this and that. And it gives rise to this. So the polymorphisms that are down here are in here, or a subset of them, and they are sorting themselves out there. Absolutely. But see, that's what I'm saying, though, is that detecting, so let's assume that this is wrong for the moment. Then you don't have this, you just have one lineage that dumped into the lake. That one lineage is going to have a lot of polymorphisms in it, right? And it could sort itself out. But the reason they know it's this hybridization to this, is because they've genotyped, they population resequenced the riverine forms, and they know that the divergent alleles from this and this are here. Okay? But that still doesn't make this a horrible kind of system for trying to draw phylogeny or gene genealogy, whatever you want to call it, or a series of them, because you have this little bitty amount of time, okay, to get in tiny little internodes amongst 450 species. So to draw a phylogeny, it's a beautiful system to look for, hey, did this cause it? Because there's a lot of other alleles that are moving around and they know where they came from. But in terms of drawing a phylogeny, it's terrible. And so, but anyway, if you will, seriously, if you want that paper, just send me an email. Yes, that's it. When was it? What was 13? Yeah, I thought it was molecular ecology, but I couldn't remember what year. But yeah. Can't remember to do a Dropbox, but I can remember one paper out of 11,348. That's pathetic. That's okay. I'm going to get over it. Okay, other questions, comments about this? This is wonderful. Okay, this is once again, sort of. Can I ask a question? I see all of you guys rubbing your arms. Are you freezing to death? Okay. Since I'm the leader here, we can turn the frickin' air conditioner down. No, I'm doing fine, but maybe we ought to turn it down. I'm just going to watch you guys go, and that'll be bad. If we know how to, thank you. Or just the fans, I guess, whatever you want to do. Seriously, if I sweat, you're paying me, so it doesn't matter. Okay, any other questions, comments about this? This particular paper, these kinds of studies? See, you guys are so nice. Americans would have shot somebody by now, okay? We all carry guns. I mean, I'm packing right? No, I'm not. I'm only kidding. All right. So any other questions here? Oh, you mean this original cross? Different lineages.
It happened differently in all.
Yeah, it's sort of like those individual lake systems in one sense up in that Dolph Schluter's looked at, except his are not, his are not many species, but it's repeated evolution. And what the other thing you need to know is different kinds of lineages contribute to the hybrid populations that underlie the explosive adaptive radiations in these cichlids. But in those lakes, this is very similar to Dolph Schluter's work. In those lakes, evolution repeats itself, right? And so, in other words, the same forms are popping up over and over and over again. Not the same species, okay, but there's convergent evolution between the lakes for different kinds of phenotypic forms, Yeah. So, yeah.
You mentioned that these fish have so many different kinds of feeding mechanisms.
Mm-hmm.
So in the other fish, sticklebacks, right? Do they not have that many different kinds of feeding mechanisms? Because they are only getting sorted into two different ones all the time.
Yeah, what I can't tell you is what the genomic underpinnings are, the regulatory sequences for, are they different? And are they keying in on, the sticklebacks do not have that kind of diversity in their feeding forms, first of all. But also I can't tell you, I don't remember if I ever knew what the genomics are. They do know what the genomics are, at least major effect regulatory elements in the sticklebacks. I can't even remember if they know in the, so I don't know if it's the same kind of regulatory element. It's probably a transcription factor, it always is. But if it's affecting the same kind of structural genes, I'm not sure. But sticklebacks definitely, sorry, the ones that Dolph works on definitely do not have the kind of diversity. But that's sort of circular for me to say. There's two kinds of forms, and so obviously they don't have 100 kinds of forms. But could they have 100 kinds of forms? I don't know. I don't know.
Why they don't have.
You know, I mean, we can go once again. I can be very circular, but I think it's a good inference. The niches aren't there. If they could diversify, then they have diversified, but there's been selection against, would be my argument. Selection against those because there's nothing they can go into, and so they go extinct. Those lineages would go extinct very rapidly. But my guess is that even if those niches are there, that actually sticklebacks don't have that kind of adaptive potential for those feeding mechanisms. It's just a guess. Okay? Yes?
System-wise, are fish more likely to have this sort of hybridization and diversity?
I would say that the examples that have been looked at suggest that no, it's not just fish, but a number of really wonderful examples have been looked at, but that's because Ola Seehausen was the author of the hybrid swarm hypothesis, and he works on fish. So he's looked at lake whitefish in Europe, and he finds very similar kind of process, where you've ended up with, in that case, glacial refugia, and then an admixture event between divergent lineages, riverine, dumping into separate lakes and sorting themselves out. And in that case, you were asking about, for the lakes, is it the rift lakes? For that, in that case, it looks like it's a common pool of variation that dumped into these different lakes in the montane lakes, alpine lakes, sorry, in Europe. But they sorted themselves out into multiple species that look very similar to one another, but it's due to hybridization, once again. But the other examples of this would be things like Darwin's finches that obviously have been driven, their adaptive evolution in terms of beak shape and size that really does drive a lot of their evolution from what people have found, what Peter and Rosemary have found, is due to intergressive hybridization of particular regulatory elements and natural selection. Okay, and that's an adaptive radiation. It's not 450 species, but it's always been considered an adaptive radiation from the time of Darwin. And then you have things like, you know, heliconius butterflies in the New World, which show the same sort of thing. They're mimetic rings, adaptive radiations, and those mimetic rings of species or assemblages are due to the transfer of a particular regulatory element and probably some structural genes in all these cases, but regulatory element that is intergressively hybridizing amongst all of those different forms, once again driving that kind of adaptive radiation. And then we have plants, where we see the same sort of plant assemblages on islands which are sympatrically diverging, and it appears like it's underlain by hybridization. So I would say that we haven't been looking for this until Ola Seehausen alerted us to it, And really nobody else was looking for it, honestly, except Ola, I would argue, and maybe some of us were writing about it. I was writing about it. But in terms of experimentation and studies of nature, it's been incredibly recent. I would say in the last 5 years, where people started saying, oh, wow. You know, like Darwin's finches, they didn't know that was a regulatory element that was being passed around. Heliconius butterflies, they didn't know that was a regulatory element that was passed around. They, you know, and so we've only been looking for this for 5 years, and so therefore, you know, the number of cases that seem to be underlain by hybridization is pretty incredible, because it's been such a short period of time that we've actually been thinking we should test for this. This is another one of those paradigm shifters, I think, for us.
Earlier, actually, following the biological species definition, people were actually looking for how the speciation would go on. So they never thought that there could be so much of adaptive integration. So now it's coming to light that adaptive integration is one major, more important than before.
Yeah, yeah. I mean, divergence with gene flow is a new moniker. You know, it's a new phrase for it, but all it is divergence with introgressive hybridization in terms of eukaryotes anyway. Divergence with horizontal gene transfer in prokaryotes, and divergence with viral reassociation for viruses. So in all of these groups, but particularly in eukaryotes, keep in mind, by the way, when we talk about prokaryotes, everybody's like, well, that's prokaryotes. Everybody knows that they exchange so many genes, blah, 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 blah. We didn't know that till about 60 years ago. All of this is incredible. We didn't know that HIV, in fact, we held that HIV could not recombine because of the kind of virus it was. And until they started sequencing it and said, good grief, how did this recombine? It can't package this way. It can't get heterologous sequences together. It's impossible. That they said it's molecularly impossible. Well, they were wrong. So a lot of these were we knew the biology, or we thought we knew the life history and biology of organisms, and we didn't. And so, but also a lot of it is genomics. It's genomically driven. People started looking at genomes and they went, good grief. There's foreign material from here, from all over. I've sequenced highly repeated sequences, or sequenced highly repeated DNA for my PhD and my first postdoc when I was in Australia from grasshoppers. That's why I know that's a lot of data. A lot of data. It took me 3 years to do a bit more than that. Anyway, I didn't know I was working on retroelements. Had no clue. And so it's the clarity that's been coming around the last 5 years is unbelievable. One thing, let me mention one, I was going to give you this example. I'd mentioned mitochondrial DNA. Keep in mind, and the fact that they used a molecule that was not appropriate for detecting introgressive hybridization, really, not very powerful, but they weren't looking for it. In our own species, out of Alan Wilson's lab again, about the same time, Becky Kahn and Alan Wilson published a paper in Science or Nature or both, I can't remember, you know, where they sequenced the mitochondrial DNA from many, many samples of us, okay? And in fact, one of my very good friends in science, he was a professor at Harvard, his baby was being born, and he was working with Alan Wilson at the same time, too, at Berkeley. And Becky actually asked him, can we have some DNA from your daughter? You know, or your child. I think they knew it was going to be a little girl. Heather was about to be born. And Rodney said, yeah, what do you need? And this gives you the idea of how the field has changed. Okay, with mitochondrial DNA, even back then, they needed a huge amount of material. And Rodney was like, I'm not going to like bleed her to death. You know, I mean, what do you need? And he said, the placenta. She said, the placenta. And so here's Rodney trying to wrestle the placenta away. I know it's sort of gross, but wrestle the placenta away from the nurse who does not want to give it to him. him demanding that this was his, and her demanding, we're not allowed to give you this, and him saying, look, I need it to DNA sequence my daughter, the nurse thinking he's a pervert, and trying to figure out what he means by DNA sequencing. So I know that's a little anecdote just to keep you awake tonight. So the thing about it is, Becky and Alan concluded that we came out of Africa, which was correct. Okay, that was accurate because there were different kinds of hypotheses, different areas. East Asia, Middle East, Africa were three possibilities from the fossil record of where we could have arisen as a species. They designated the root was in Africa, period. It was very strongly there, and we now accept that. But they also said we came out of Africa 50,000 years ago, 100,000 years ago, whatever it happens to be now. And when we came out of Africa, we did not hybridize with anybody else. We have no weird mitochondrial DNA in us. Well, once again, that stood until 2010, when they had genomic information from Neanderthalensis and Denisovans. So all of us who were arguing, it's almost certain that we hybridize, and here's some genomic information that other people have from the nucleus, and everybody went, Becky Kahn and Alan Wilson showed in 1990, maybe 90, I don't remember when it was, that we didn't hybridize, and we didn't hybridize. And they were using non-appropriate data to test for that. Okay, so I mentioned I was going to give you another example of why these kinds of data, non-recombining molecules, plants, chloroplast DNA, animals, mitochondrial DNA can mislead us, can give us a non-test for admixture. Okay, yes?
Whenever you call a population monophyletic, how do you decide that how much what you want to do?
Yeah, so what I'm guessing you're asking, let me make sure I understand. So if we have, so are you asking how far down we go this way? Yeah, so you could call this, in some ways, we could think of it as being multiple ancestors, right? But that's, in terms of cladistical terms and phylogenetic systematics terms, these are all monophyletic because between these nodes is one ancestor. So regardless of how far down you go, all of this is considered a monophyletic assemblage. At any point in time, that's just the phylogenetic nomenclature. But I understand your point. If we cut it here versus here versus here, is everything above that or below that still monophyletic? But it is according to phylogenetic systematics terminology consideration. It's just that what we're saying is, by this, is that you have the calving or the cladistic, you have a cladistic event, each one of these, but it's from a single ancestor. Hence the internodes. But down there it's not. So whenever we have, for example, an allopolyploidy event underlying a clade, that entire clade is non-monophyletic now. Now, some would say, no, it's not. I mean, this is non, maybe, this is polyphyletic, and that is, but everything else is monophyletic. Well, yeah, but they have multiple genomes from divergent lineages. So by definition, first of all, Willy Hennig in his Phylogenetic Systematics book in '66, whenever it came out in German, argued that that was impossible. You could not have this phylogenetically. It was impossible to have the origin of a lineage from two lineages. He said that was just phylogenetically impossible. And I think I mentioned this, that it's an ironic argument because he said you count the number of tips. But what he said was, he said the reason this is impossible to arise, for a new species to arise from this kind of an event, is he said we know different species are reproductively isolated. So he used a non-phylogenetic argument. He used a biological species concept argument. Okay. Great questions, though, once again. Easier since you had the readings? Jeez, I'm never going to get over that. Any other questions on this or these kinds of topics? We can move on, spend a little bit of time talking. We probably won't finish it because I want to talk through the Scott's work on Columbines. Any other questions on this? We can come back to this as you think of other things. Yeah.
Is there any evidence for multiple hybridization?
Is
In the lake case,
uh-huh.
is there any evidence for multiple hybridization rather than single event happening in the area?
Where are you talking about? Up here?
Yeah.
Yeah, there's introgressive hybridization. There's homoploid speciation within the lakes after the divergence.
Does that happen multiple times over time?
It's happened. Okay. Here's the problem. Once again, before I say this, is people weren't looking for it, and they remember I said the photo on the front of my book. One of those is a homoploid hybrid species that Ola Seehausen's lab has defined. And they've looked at how it would form in the lab and all this sort of thing. For the most part, people were not looking for homoploid hybrid species following the invasion and diversification. However, even without looking for them, they have detected a handful, okay? We don't know what the extent yet, and I've asked Ola and Axel and others, and Walter Salzberger, who also works on this group, who's in Switzerland. I've asked them, how many homoploid hybrids are there? How much integration is there? And they said, well, there is assortative mating among lineages, but we know there, in some cases, extensive introgressive hybridization going on among multiple lineages, and we are starting to do the kind of work to test for what the frequency of homoploid hybrid speciation is. So far, they've seen a handful of detected lineages that came from lineages like this, hybridization events, not allopolyploid, but hybridization events. And so it's going to be more complex, once again, as they get more genomic and resequencing information, it's going to become more and more complex. They're convinced of that. But also, just because it's increasing how many cases they're finding. So I can't give you out of the 200 or 450, are there 30 homoploid hybrid species? There may be more, maybe fewer. But they do know that there's ongoing introgressive hybridization and that it's affecting adaptive traits like sexual selection kinds of things, but also ecological traits. Yeah?
Not related to this paper or current discussion, but I remember you mentioned a couple of times like humans are polyploids.
Mm-hmm.
So could you explain how do you mean that? What do you mean by that?
Oh, can I explain what I mean by our genome being possibly at least six-fold whole genome duplication, so 6x, but it's likely we're 8x at least, so we're octoploids. So what do I mean by that? I don't mean that since the primates formed, or since mammals formed, that there have been whole genome duplications, although there have been in some lineages, likely, but rather that as the vertebrate lineage diverged from the invertebrate lineage, several whole genome duplication events have been detected. Same thing for plants and fungi and all organisms. We have whole genome duplications in everything. Not necessarily prokaryotes, obviously, and viruses, but so we also have a lot of segmental duplications. But in terms of whole genome or polyploid, what's referred to now as WGD, whole genome duplication events, we've had a whole series in the vertebrate lineage. And that's why we can say our genome and those of all other vertebrates. See, bony fish split off and there was a whole genome duplication event on the other side of that that we don't share with them. So there are additional whole genome duplication events in vertebrates that maybe we don't have, but that's what I meant. And the way they know that is, once again, looking basically for duplicated loci, extensively duplicated loci across, many duplicated loci across the genome, and asking whether or not it's more likely segmental duplications, because we know those are quite frequent, or is it more likely it was a whole genome duplication? And as they add more more genomes of key species, key particular species in the internodes, that's when they are able to test it more and more rigorously as to how many whole genome duplication events there are. They know there are multiple, but as I say 8x, for 6x, that's still an enormous amount of duplications, obviously. Once again, shocked. We were all, it was like, but that doesn't happen in vertebrate lineages. How can that be? Well, it does, by the way. Amphibians, reptiles, so we have a lot of neopolyploids in the vertebrate clade, even today. And there's one that's been hypothesized for mammals even, a neoallopolyploid event. Actually, a couple of them in some rodents in Patagonia. And that one is, it's actually one of my very good friends was a co-author on the first Nature paper on that. But that one is being examined by other people, wondering if it really is a whole genome duplication or not. So that one's up in the air. But there are neopolyploidy events all through the vertebrate lineage now, or vertebrate clade even now. So ongoing whole genome duplication events in a number of clades. Okay, other questions or comments? How are we doing? Okay. One more question, and then if you have it, I think we'll go ahead and, instead of me getting 10 minutes into the discussion of this next paper, I think maybe what I'll do is ask if we go ahead and do our break. But any other questions before we take this break? Yeah.
Very general question. So why is allopolyploidy so much more frequent in plants than animals?
So the question is, why is allopolyploidy so much more frequent in plants than in animals? What you're referring to presumably is neo-allopolyploidy, like, recent allopolyploidy, but maybe even more ancient. So here's my problem with answering that is actually even accurate, is we just talked about all of vertebrates have numerous whole genome duplication events marking our genomes. Many lineages of current vertebrates have that. Some lineages have neo-allopolyploidy going on. Some clades don't seem to at all, like mammals, for example. But even those mammals have, octoploid or whatever genomes. So is it less frequent in animals? Neo-allopolyploidy, I would argue, is probably less frequent in an animal clade than in plants. But if we talk about evolutionarily effective, the frequency may not be any higher in plants. It's ongoing still in plants, but then it's ongoing in amphibians and reptiles and other kinds of animals. But what I mean by that is we know the frequency of neo-allopolyploidy in angiosperms. It's fairly low, right, compared to the number of species that are out there, and yet every angiosperm is an allopolyploid. In other words, if it's always been relatively infrequent, as we move up a tree, it's always been relatively infrequent in terms of how many neo-allopolyploids are being formed, throughout that evolutionary history of angiosperms. And yet we get up here, and every one of them is at least an ancient allopolyploid. So somehow those very infrequent allopolyploidy events in these lineages transferred themselves to incorporate or throughout every clade and every lineage. Now we could say, well, it all occurred here, that's why. But it didn't, right? I mean, a lot of these allopolyploid events, so this whole lineage was not allopolyploid until there, or this whole clade was not allopolyploid until here. So somehow the infrequent events are very evolutionarily, they're captured by these organisms. Same way with the vertebrates. Why are we octoploids or heptaploids or whatever, as humans and other mammals? Why were those events so evolutionarily successful that they're transmitted all through that clade? Why didn't they go extinct? And so I rather turn the question that way and say every vertebrate is a multiple, probably allopolyploid. And every invertebrate is whatever, and every fungus is whatever, and every angiosperm is whatever. And so I'm not sure it matters that it's less frequent in animals if we're all allopolyploids already. In other words, currently it's less frequent, but maybe it pops up infrequently and then it's captured by the entire clade. Okay, so that's that little spiel, but there are reasons why we would think that it would be difficult for you and I and our partners to produce non-reduced gametes now and produce a progeny that is allopolyploid. And one of the major reasons that we would, and that's within a species, I realize, but allopolyploidy is considered to be possible within a species too. And there are reasons people talk about gene dosage in vertebrates and animals in general, but it has to be not even just all animals or vertebrates, right? Gene dosage, and also what about the sex chromosomes? And so having unbalanced or too many or too few sex chromosomes of different cases in heterogametic organisms with heterogametic sex chromosomes is known to cause problems, developmental problems. So those are some of the suggestions, is that organisms with sex chromosomes do not allopolyploidize. Organisms that have certain kinds of developmental pathways don't. And most of the organisms they point to, obviously, are animals. But we actually know that organisms with sex chromosomes, and amphibians and lizards and snakes actually do have lots of allopolyploids. So it's not a completely definitive answer at all. But so people have studied this, and there are hypotheses, but we don't know why it really gets shut down. We know it appears to, but yeah. But just keep in mind, this is very important distributed kinds of events evolutionarily somehow that get into all the lineages that we look at, probably all plants, not just angiosperms, and all animals have whole genome duplication events in our evolutionary history. Yeah.
Is there any link between presence of stress and allowing such events to happen, where you would otherwise reject it?
In terms of like allopolyploidy?
So allowing it to, for example, you might have certain defenses against the formation of alcohol, but then in terms of stress,
Mm-hmm. What I can say in terms of environmental dependent stress or whatever resulting in these kinds of events is that it's possible, and we know that spindle fiber formation and that sort of thing can be disrupted. So we're talking about in meiosis, right? We've got to get unreduced gametes if we're talking about polyploidy. By definition, you have to have diploid gametes and diploid gametes meeting up with other diploid gametes to get a tetraploid. So what we know of in plants and in animals too, our own species, is that we produce a certain frequency of gametes that are unreduced. What we don't know is exactly the genetic or the environmental situation of why that can be. I mean, there are chemicals that do it. So maybe it's just exposure to us. You know, we get exposed to certain kinds of spindle fiber disrupting chemicals, or plants do. But it could also be genetically predisposed, where you actually have the actins and et cetera, that function of those genes just occasionally gets switched by a mutation or something in producing those gametes. But we really don't know. As far as I know, we don't know yet. And I may just not know the literature well enough, but I try to keep up with that. But yeah.
I experimentally it is very easily possible to get allopolyploids in plants using chemicals and everything.
Yeah, you can, yes, we can produce neo-allopolyploids in plants.
Same kind of endpoints are not exactly possible with animals given formulation.
Well, we can get, well, those experiments have been done in certain animals, and it is possible to get neo-allopolyploids in animals, but they only develop usually, okay, once again, enormous numbers of lineages in amphibians, snakes, lizards, etc. are autopolyploids or allopolyploids, usually allopolyploids. Okay, so understand that this happens in nature in animals. We have to broaden our scope away from mammals, basically, and birds, right? Because we focus on those kinds of, well, I don't study amphibians or whatever. Well, I don't. But, you know, so we have to broaden it and realize that there are whole clades that are marked by many, many of these kinds of events. So it's not simple enough to just say we can't do it in animals. However, in those animals that seem like they don't do that, if we try those experiments, yes, we can get some kind of start to development, but they fail, right? They're inviable in the cases where it's been tried. In those animals that don't seem to throw those kinds of allopolyploids off, form them, I mean. Just understand, tons and tons of frogs do this, tons and tons of lizards do this, and tons of snakes do this, in terms of numbers of lineages. So we need to keep that in mind. This is actually a quite frequent event in certain kinds of animals. Yes?
Curiosity. So we have some genera in plants which are quite specious,
Mhm.
say orchids. So does that have to do anything with the Lloyd level, and that is why they are?
I should know this, but the orchids, okay, initially the orchids are thought to, like figs, okay, like ficus, are thought still to be driven by pollinator selection, right? So one pollinator, one orchid species. I have my doubts about that. I've always had my doubts about that, but now the ficus example shows pollinator sharing. They just published a paper, just came out in Proceedings of the Royal Society. And so they're demonstrating pollinator sharing, and they also, with their genomics, are demonstrating that integressive hybridization is quite widespread among figs. My guess is that's what they're going to find with orchids as well. So that's the first thing I would like to say. The second thing I would like to say is I can't remember what orchids have in terms of polyploidy. You need Mark Chase here from Q. Mark would say, Mike, you should know that. And I would go, I know. But there's a lot I should know, but that I don't remember. My perception or my really hazy remembrance is that they do not have the levels of polyploidy that you see, allopolyploidy and autopolyploidy that you see in other plant groups, some other plant groups. But that is, I really need to check that to confirm what I just said to you. But that's my remembrance. Okay, well, why don't we go ahead now and take a break since it's 11. Thank you, guys. This is fantastic, okay? If I don't show you a PowerPoint slide all day, I love it. All right, but I may still do that. So